So what types of cells exist? So you can kind of break this up into many different things, okay? But here we can look at sort of a tree, and we've got, of course, the group Eukarya. These are our eukaryotes. That includes all those nice fungi. We've got animals, amoebas. Amoebas are very cool. Land plants. We really don't give these guys enough respect, okay? So I'm saving a lot of that for chapter 10, but I'm telling you, okay? We've also got some extra interesting types of things, some algae, diatoms, always neat to look at. Bacteria will probably be the other place where we spend a lot of time in cell biology. Lots of different types of bacteria, okay? Again, we'll be kind of hinting at some foreshadowing here. We'll be talking about mitochondria and chloroplasts and sort of how they're related. Okay. Archaea, we won't spend too much time talking about them, but I may mention them now and again, particularly things like maybe sulfur-producing bacteria when we get to that unit of metabolism. So that would be chapter 9, okay? Meet things like thermophiles living in thermal vents, halophiles, high salinity areas. There's just a lot of really cool diversity here. But we're going to be spending most of our time with animal plant cells and some bacteria. So just to get us going, let's talk about what are some common elements of all cells from all categories. Okay. All cells are bound by a plasma membrane that's going to be composed of a phospholipid bilayer. So we're going to be talking about more about phospholipids in chapter 7. These phospholipid bilayers also have proteins of various compositions that we will touch on a little bit later, but they're all going to be kind of encased in this plasma membrane. The space in the plasma membrane is going to be filled with cytosol, also called the cytoplasmic space. Okay, the cytosol is kind of a fluid. It's going to contain things like water, small molecules, things like sugars that we're going to use for metabolism and building blocks. We're going to have nucleotides to make DNA, RNA types of things, enzymes, other proteins, all kinds of stuff. So this is all types of cells. And they're all going to contain DNA as a genetic material. So we've got these things in common. We've also got ribosomes that are going to synthesize our proteins. So we've got a lot of things in common, but we also have a lot of differences. So some differences between prokaryotic cells and eukaryotic cells. So prokaryotic cells, which are going to be our bacteria types of things, the hallmark is that they do not have a nucleus or any other internal membrane structure. So they've got the plasma membrane and that's it. Everything else is sort of like dumped in here together. Even though they have common elements, they're not organized. This is just like all sort of dumped into a space. They're largely, on the whole, smaller than most eukaryotic cells. So most are about 1.5 micrometer in diameter. You can find even smaller, but these, in general, can be smaller than most eukaryotic cells. Their DNA is structured a little bit differently. We'll be talking about this when we get to chapter 16. But they have circular DNA. So they don't have nice linear chromosomes like eukaryotes do. They have circular DNA. Okay. Their DNA, instead of being in a nucleus, is more what we call a nucleoid region. They also have a cell wall, which can be composed in a variety of different ways. This is where you get this sort of gram-negative, gram-positive idea coming from their cell walls. Okay, which is something that, aside from plants, we're not going to see too much of in the eukaryotic world. Okay, eukaryotic cells are really kind of highlighted by the fact that they have membrane-bound structures on the inside. They have nucleus, they have all kinds of things like mitochondria, they've got endoplasmic reticulum, Golgi apparatus, which we'll be talking about in the next couple of videos. Okay, they've got a lot of things going on. They're, in general, larger than a lot of our bacterial cells. We have linear chromosomes on the whole, so that, again, will be a little bit different than what we see in prokaryotes. and no cell wall in our animal or protozoan cells. So we know that plants do have cell walls. Okay. So there are going to be some advantages associated with compartmentalization. All right, this could include things like being able to separate out transcription and translation. This will be things maybe even like separating out different processes and enzymes in the cell. So you'll have some questions as part of your pre-lecture material, and it may ask you to think about what are some advantages that you can think of that are associated with compartmentalization. So now that we know a little bit more about the composition of cells, things that they have in common, things that are different, we can also think about why cells are the size that they are. So why are cells larger than viruses? What defines the lower limit? So if you think about a virus, we're going to effectively have just sort of a protein structure, sometimes some lipid membrane around it, an envelope, but really all that's inside it is genetic material, right? So what are some things that cells have to fit in order to do their job? They have to have ribosomes, they have to have at least some type of enzymatic functions. They've got to have proteins that are going to be rolling around in there. So they can't get away with having as little material as viruses. Okay, so that's why our cells have a lower limit. Okay, so what about the upper limit? Why are most cells so tiny that we need a microscope to see them? In order to understand this, we can imagine a cube with an arbitrarily determined length and width of unit 1. Okay, so there's just one unit, arbitrary units. Its surface area is going to be the sum of the area of each of its 6 faces. So we've got our surface area, number of faces, equals 6 arbitrary units squared. Okay, so keep that in our minds here. I'll leave it listed. The volume of this cube is going to be the length times width times height, okay, which gives us length with height, 1 unit cubed. Okay, so this gives us a surface area to volume ratio of 6. So surface area to volume ratio is 6. Okay, so what we're going to be looking at is the surface area to volume ratio, and let's just say that the surface area to volume ratio 6, we'll define that as the limit to stay alive. Okay, and why is this ratio so important? Cells need to do a lot of things at their membrane. Okay, they need to take in oxygen. They need to get rid of carbon dioxide. We're talking about metabolism. Okay, cells must also be able to take up nutrients from the environment. Okay, so oxygen in, carbon dioxide out, nutrients in. Cells need to be able to do this. Okay, and they need to be able to diffuse this throughout the entire volume of their cell. So additionally, some cells are specialized to absorb or filter molecules from the environment. Okay, so you can also think of the structure of like intestinal cells. They have to absorb things from the environment. Okay, so they often have projections that allow them to have more surface area. Other types of structures are going to have important enzymes or receptors embedded in the membrane that we need more of. Okay, so these types of cells have specialized shapes made to maximize surface area. All right, so here you can see structured intestinal cells. You've got these projections. Neurons need to be spread out quite a lot. Okay, so increasing sort of that surface area to volume ratio. Okay, so we're going to again define the surface area to volume ratio of 6 as the minimum threshold for life. Anything under 6 and it's bad biscuits. Okay, so now we know more about that. Let's imagine something larger. So here's our old cube. Now let's try a larger cube that's 6 units long with 6 units high, 6 units deep. Okay, this cube has a surface area of 216 units squared and a volume of 216 units cubed. Can it stay alive? No, because its surface area to volume ratio is only 1, and we have defined the minimum for life as 6, okay? But we obviously have things on this earth that are larger than a single cell. So how do we achieve this? Let's divide our box into smaller units. So it's still overall the same size, but smaller. Each of the smaller boxes that we see within our larger box has the same dimensions as our smaller box from before. The total combined surface area that we have now, including all the edges that we can't see here, okay, is now 1,296 units squared. But its volume is still that 216 units cubed. So now our surface area to volume ratio is 6, and we can stay alive, okay? So we've learned more about some of the constraints that govern cellular structure, okay? How big they are, how small they are, some of the things that they have in common, and some of the things that are different between eukaryotic cells and prokaryotic cells. Okay, so I'd like to end today's video just looking at and appreciating the beautiful diversity of cells. So we've got a lot of different types of cells here just for reference. If you're looking at the PowerPoint, you can find the labels in the notes part of the PowerPoint. Okay, we've got some fungal hyphae type cells here. All right, this is going to be a spirochete type bacteria, red blood cells and different types of blood cells, platelets, white blood cells. Okay, this is human sperm and egg. All right, just a lot of different things. I believe this one is a retinal neuron. This is a xylem. Okay, intestinal cells with those projections. All kinds of things that you can see. I think that this is a radiolarian and some type of protozoan, but you can see that they have structures that are going to be similar to their function. Sperm, you can see, they're designed to move. They've got these long flagella that help them move. Retinal neurons have to make a lot of connections, so you can see the way that they are branched out. Intestinal cells, they're there to absorb nutrients, so we got to have a lot of surface area in order to be able to do so. These spirochetes, I don't know, I never really met a spirochete I liked, so I think they're basically shaped this way in order to screw us over. So that's kind of my opinion about spirochetes. Fungal hyphae, again, we're just trying to get into the environment to spread, get nutrients, grow through the environment. So you can see a lot of structure, function type of relationship. So just to finish up, there are many different ways for us to study cells, many different types of microscopy. We've covered a few types of light microscopy and a couple of types of electron microscopy. The way that we want to be thinking about this is that the tools that we use depend on what it is that we're trying to achieve. What do we want to see? So we want to use certain types of microscopy in certain situations, maybe another type in another situation. There are also many different types of cells, and while they share the same basic characteristics, they also have different features that are highly specific to their functions.